Hey guys, my name is Chelsea Lopez. I am a first year master student. This is actually not my first master's though. This is the second master's that I have or will be completing. My first is in agricultural education and communications from the University of Tennessee. However, this master's I will be doing completely or predominantly online as I am an agricultural and natural resource extension agent in Southwest Georgia. So I'm actually about 3.5 hours away from campus, and therefore you guys have not obviously seen me in class and probably won't. But today I am going to go ahead and talk to you guys about milk synthesis and secretion. I want to go ahead and make a forewarning that I do not have a background in biochemistry. It certainly is not my strong suit, and there is a lot of rabbit holes that you can definitely go down when it comes to milk synthesis and secretion. So I did my very best to explain everything to the best of my abilities, and hopefully it will make sense to everybody. So we'll go ahead and get started. Okay guys, so I just wanted to go ahead and start with going over the basic anatomy and the purpose of the mammary gland. Obviously I think we all know what the mammary gland is and what it is used for, and therefore know the importance it serves in milk synthesis and secretion. So to go ahead and start, the mammary gland begins to develop when it's in its embryonic stages. So when it is a fetus, it does begin to develop. This development at an embryonic stage typically is just very basic development. It'll have basic epithelial, and then we will start to see around 75 days gestation, we'll start to see that mammary gland appear. And so again, for the sake of this presentation, everything and the days and the information I'm referring to pertains to our ruminants, obviously, which is what we are doing in class. So at 6 months, when the fetus is 6 months, we will start to see a pretty much fully developed udder. It's going to be a very basic developed udder, but nonetheless it should be pretty developed at 6 months of age. And so after birth, they will begin to grow at a much quicker rate. Those mammary glands will begin to grow at a much quicker rate, which is what they typically call allometric growth. And so there have been several studies as to why all of a sudden at birth this allometric growth happens, and it's believed that it could be linked to ovarian function. And so again, we tend to see a lot faster growth in those mammary glands after birth. So the mammary gland itself does consist of 4 separate glands. Those glands have connective, fibrous, and secretory tissues. And because it has 4 separate glands is why we will never see milk that is synthesized in one gland pass to another. So the right and the left sides are separated by a ligament, and then the front and the rear are also separated. So you'll never see milk that's synthesized in one gland be passed over to another gland. And the mammary gland, obviously, as we all know, is designed for suckling and therefore is influenced by pressure and heat, and as well as the frequency of suckling. And so sensory nerves within the skin of the teat become sensitive to that suckling, and which is what kind of initiates that lactation, which we'll get into a little bit later. So this picture on the left-hand side, you'll see each figure of embryogenesis, puberty, gestation, lactation, and involution. And on the right side of the right half of each of those little pictures is how the mammary gland continues to develop through each of those stages. Okay, so as we then or now get into the development or the specifics in the development of mammary glands, I'm going to take a step back to again talking about what happens in the fetus. And so those tissues that will eventually become the connective tissues in the mammary gland, they're signaled, and it triggers the epidermal cell embryo to yield these epithelial cells. And this happens, and this is going to be where the future mammary glands are going to be. And so this process, as complicated as it sounds, so basically just a bunch of signaling for connective tissue to tell the epidermal cells to make epithelial cells, is going to continue to happen through the fetal development. So as I said, we see at 75 days gestation that the mammary band will start to develop, and that around 6 months we will start to see a fully developed or an almost fully developed udder. So this process is taking place during that time, building up those mammary glands. And so when we fast forward to birth, and again, remember that that process that I just explained above happens at a much slower rate than when it'll start to happen once the calves are born. And so once they're born, it's tend to believe that they begin what we refer to as allometric growth, which is just that growth happening at a much faster rate. So at birth, there is an epithelial or basic epithelial rudiment at the site of the teat that has a mixture of stromal cells. And so the mixture of those stromal cells are adipocytes, fibroblasts, immune cells, and primordial cells. So we'll see that if you look at a calf just born and you were to take histology of the teat, and look at that under a microscope, those are probably some of the cells that you're going to see. And so that epithelial parenchyma is going to be arranged in an immature set of ducts. So it's not going to be a very defined pathway, or set of ducts. It's just going to be a very immature, but they're going to be there. And so that's going to be at the base of the teat. And that all happens by a process called alveologenesis. Sorry. A lot of letters, big words. And so that process, if we break down the word, is basically just the development of those alveoli that we'll later talk about and are pretty critical in the process of milk synthesis and production. And so that alveologenesis is induced by progesterone and prolactin. And so it's important to know that a cow's milk production can very easily be influenced by the number of secreting cells and secreting tissue that she has. So if we look, or if we think about it from the perspective of, we see dairy cows, specifically Holsteins, that have really big udders. A lot of the times we think, wow, she produces a lot of milk, but that's not necessarily the case. Just because they have large udders doesn't mean that they necessarily have a lot of secreting cells and or secreting tissue that's going to allow them to produce an excess or a lot of milk. And so bigger udders doesn't always mean that they're going to produce more milk. Those large udders could solely be excess connective tissue and fibrous tissue and things like that will never aid her in producing a lot of milk. And so just a little tidbit, there was a study in 1987 when we talk about how fast this development happens and when it happens. I found that there was a study done in 1987 where it was thought that all allometric growth in heifer mammary glands would begin around 2 to 3 months of age. And if we think about what happens at that time period now, that's right about when we're going to start weaning calves. So in 1987, they thought that mammary gland was increasing in its growth rate right around the time that it was weaned. However, since then, there's been studies that suggest that at 2 to 3 months of age, so when we're being weaned, their mammary glands are well into active growth. So they're not increasing necessarily in their growth rate, but they're already well into their active growth in developing those mammary glands to get ready for, lactation and things like that. And so, little fun fact, cattle only have one single galactophore, and that helps drain each teat, while humans have 8 to 10. Rats, different animals, things like that, they tend to have different numbers as well. But humans, we have 8 to 10, but cattle only have one of those galactophores that will drain each teat, will drain the milk out of each teat. Okay, so as I mentioned on the previous slide, through alveologenesis, they develop what is called the alveoli, or alveoli. And so these, as you can see on the left-hand side, these little bulb-looking things are the alveoli. And so within the alveoli, there's several different structures that we'll kind of discuss a little bit later. But with the alveoli, milk is continuously synthesized in the alveolar area within the secretory cells. And so the milk is then stored within the alveoli. So again, this whole structure is where the milk is stored. And then it's also stored in the milk ducts, the udder, and the teat cistern. So we'll get into that too in a little bit. So 60 to 80% of the milk is stored in the alveoli and small milk ducts, and about 20 to 40% of it is stored in the cistern. So we see a lot of milk storage here in the alveoli. So it's important, and that'll tie into what we talk about a little bit later. It's very important that these alveoli stay active and stay healthy because they are the main things that are producing or synthesizing that milk and helping it secrete into the milk ducts, down the cistern, and through the teat. So alveoli range from anywhere from 50 to 200 millimeters in diameter. And so they're within a lobe, which is the photo that was on the last slide. That photo was of a lobe, and as you saw, there were several of those bulb-like structures on one lobe. So the number of alveoli that you'll find on a lobe is different. There is no set number. So if we kind of break down the internal structures that we've talked about, or some of the structures that we've talked about already that are within the alveoli, as you can see on this picture here to the left, the one that's cut open and kind of facing towards us, those square things that run on the internal part of the alveoli and have the little dots in them, obviously you can see there's an arrow pointing to them. Those are the secretory cells, and so that is what is going to actually secrete that milk within the alveoli. That big wide open space is called the lumen. So when they talk about where milk is actually stored, it is stored within the alveoli, but it's actually stored within the lumen that's within the alveoli. And so on the outside of that alveoli, you definitely have some capillaries, because as we'll later find out, a lot of the milk components, nutrients are supplied via the bloodstream, and so there's a lot of capillaries that run through it, and so it's very important for blood flow to be consistent and to be proper through the mammary gland. And so if you look over to the picture, or that is on that same picture, but the structure that's on the left, it is the same structure, just looking at it from a different perspective. And so you can see that the basal membrane kind of surrounds it, and then you have those myoepithelial cells on the outside of that alveoli. And again, capillaries will, surround that, providing blood flow and nutrients to that alveoli. Okay, so this is really just something that I felt a little important to include so we can kind of understand the mammary glands and how they actually work, and not necessarily how the milk is synthesized and secreted, but just the importance of mammary gland function. So, as I had mentioned before, the mammary glands are heavily supported by blood vessels, arteries, and so it's important that the udders have good blood flow. And so each udder half has its own arterial supply. And so this anatomy, with their own arterial supply, has the intent on providing those, that half of the udder with continuous supply of nutrients to those milk synthesizing cells. Sorry, I had a brain fart. So if you look on the left-hand side, this is kind of a very brief overview, and I'm sure I could have gotten into a whole other rabbit hole of the vasculature system that goes into the mammary glands. But this is just showing you kind of the blood flow that, of how the blood flow comes out of the heart and goes to the mammary glands, and then versus the blood flow that's coming away from the mammary glands and back into the heart. So just kind of shows you how that flow happens. And so again, that blue line is blood being pumped away from the udders, and then the red line is the blood being pumped to the udders. And so you also see here on the right hand side, this is just a very brief diagram of kind of the lymphatic system that supports the mammary glands. Again, it's important to make sure that we are taking away any waste from the mammary glands, and so those lymph nodes and that lymphatic system is definitely responsible for doing that. So they do have their own, udder lymph nodes and things that help support those functions. Again, another little fun fact, in order for a cow to produce 1 liter of milk, they will need their udders to be supplied with at least 500 liters of blood. So if a cow makes about 60 liters of milk a day, we're talking about pumping around 30,000 liters of blood through that mammary gland. Okay, so now that we've gone over a little bit the anatomy of the mammary gland, which again, just a little bit of an overview of that is, it starts within the fetus, continues through birth, and then at birth, that mammary gland rate will start to increase in its growth rate. So it'll start to grow and develop a lot faster, but at 6 months of age, we will start to see, at 6 months of fetal age, we will see a almost fully developed udder. At 75 days gestation, we will see the mammary band start to develop. But again, those connective tissues trigger the epidermal cells, embryos, to put out those epithelials, and those epithelial cells will then start to build that mammary gland. And again, we have the epithelial cells that build those alveoli, and those alveoli then have those secreting cells inside, as well as the lumen, and that's where the milk is stored. So as we kind of get into a little bit the actual process of milk synthesis, it's important to realize that milk synthesis is not going to continue if milk is not fully or not effectively removed. So I'm sure that there are several people here that have heard of mastitis. Not technically what happens if you don't, if the cow doesn't dry up and if the milk is not effectively removed, but that is one of the things that certainly can happen. But there's several other infections and discomfort that a cow can have if milk is continued to produce but not removed, and eventually that process is going to stop at a certain point if that milk is not removed. So those milk secreting cells that are within the mammary gland, again, those secreting cells is what we refer to them as. they're within the alveoli area. They are continuously providing nutrients to that alveoli and to those secreting, um, to those secreting cells from the bloodstream in order to make what we will then see as these milk components. So again, the secreting cells in the mammary gland are continuously provided with nutrients. The secreting cells are within the alveoli area, and their nutrients are coming from the bloodstream. Some of the hormones that are needed in order for this process to happen are prolactin, again, estrogens, and adrenocorticotropic. I'm not even going to say that I know how to pronounce that. But anyway, so those are some of the hormones that are needed in order for that to happen. So anyways, once those nutrients are provided to those secreting cells, then they will start to make those milk components. And so those milk components are synthesized within the cells, specifically within the endoplasmic reticulum and its attached ribosomes. So the endoplasmic reticulum and the ribosomes get the energy by the mitochondria in order to start synthesizing those milk components that are coming from the nutrients within the bloodstream. So again, we're starting to build a little bit of a process here, and I promise it only gets longer. So then those components are passed along the Golgi apparatus, which will be responsible for moving components out of the cell in the form of vesicles. So I really wish my little circle video head was not right here in this corner so that you could see the fun little illustration that I found. But basically, somewhere right behind it, there's little purple dots, and so those are the milk components. And so those milk components kind of go in to the top part of that secretory cell, and you'll see how they're then turned into these little purple dots with a black ring around them. So that basically is that vesicle that it's talking about. So within that vesicle are several milk components, but it needs to be in that vesicle to actually be able to make it back out of that secretory cell into the lumen as milk components, whether that's fat, proteins, etc. So in order for that, again, process to happen, so you have this whole secretory cell here, the nucleus, the endoplasmic reticulum, and the mitochondria that's supplying the energy to the ER in the Golgi apparatus to form those vesicles around those milk components. So within those vesicles, there are aqueous non-fat components and lipid droplets. So also in that photo, you'll see these big blue blobs, and so eventually these become, and you can also see it up here at the top, on the top photo, they become these fat droplets. And so these fat droplets will then, eventually be able to make their way out of the secretory cell. They'll be pinched off, and that's actually going to be the next thing. So let me get to that first. So vesicles must pass through the cytoplasm and the apical plasma membrane to be deposited into the lumen. And so the plasma membrane is continuously renewed by those vesicles. So again, those little fat droplets that come in and are made by the milk components, they will then be released out of that cell and be released into the lumen. And so that's how milk components come in, whether that's via the diet or, the free-floating or undigestible proteins and things like that come when an animal eats, and or amino acids that are in the body. So all of those are technically become milk components at some point and make their way into the secretory cell and are eventually engulfed by a vesicle. And that process happens in the endoplasmic reticulum and the Golgi apparatus, the mitochondria supplies the energy for that to happen. And then there are fat droplets that are also pinched off in the secretory cell and released into the lumen, which gives our cows the fat content within their milk. Okay, so now I'm going to go ahead and just talk a little bit about lactogenesis and Galactopius, or Galactopius. That's what we're going with. So lactogenesis is the initiation of lactation, and the anterior pituitary produces the hormones that help with lactogenesis, and so that is the big hormones that we're going to see with that are oxytocin, prolactin, also some glucocorticoids, and a couple other things, but again, not going to get too much into the hormone side of things because that is an entirely different rabbit hole that I could have gone down. That would have been another hour-long presentation. So anterior pituitary glands are very important for producing the hormones that help the process of lactogenesis start. Again, lactogenesis is that initiation of lactation, and it's the hormone control of lactogenesis is not completely understood. There are several studies or several theories that they have as to why, how those hormones control lactogenesis, and some of those are the rise in blood levels of prolactin and the adrenal glucocorticoids, and that along with the drop in progesterone and glucocorticoid binding with globulin at parturition could be how that happens, how lactogenesis happens. And then, so again, oxytocin is very important, is a very important hormone that begins that milk letdown process. And so when that milk letdown occurs, the alveoli begin to compress, causing pressure within the udder, which then causes those milk components that were stored in the lumen to be released into those milk ducts. So as you can see here on the right-hand side, you have those secreting cells, again, which we talked about. Once those milk components come in, they are pinched off and stored in the lumen. And so once that oxytocin is released within the body, these alveoli, the whole alveoli will begin to compress and then force those milk components down from the lumen into the milk duct. And so from the milk duct, it travels into the cistern, and then down the teat cistern, and then through the teat canal. And so when we talk about the Galactopius, that is just a term, a very difficult to pronounce term, that just refers to the maintenance of lactation. And so again, in order to maintain lactation, it is heavily dependent on milk being removed. Okay, so now the moment we've all been waiting for, because we all just love, live, and wake up breathing nutrition and just dreaming of nutrition. And that's what we're all here for. So this is a graph that we all should be probably really familiar with, and stuff that we all already know. So we know that that our ruminants are going to ingest non-protein nitrogen, obviously some feed proteins, fats, carbohydrates, and how those end up getting absorbed and digested and made into feed proteins, fats, sugars that turn into glucose that end up being used within glycolysis and all of those other pathways that we've learned about thus far. Those fats that get turned into fatty acids, and then can also get turned into glycerol that again is used in several metabolic pathways, and then those proteins that can get turned into peptides that then make amino acids. So this is just a very brief overall graph of, again, things that we already know and how the feed components are broken down into things that the body ends up using. So now I'm going to take that chart and I'm going to take it a step further, and we're going to look at how all of those feed components, how they're digested and end up becoming milk components. So when an animal intake, the crude protein that's in a feed, that gets in, that eventually gets turned into, or can get turned into, some amino acids that go into building the milk protein. So we'll talk about that a little bit later, but those amino acids are the building blocks for the proteins, the casein and the whey proteins that we end up seeing in milk. And then, of course, we know our sugars and our starches, they get digested, fermented, go through glycolysis, turn into pyruvate, can go through the Krebs cycle, and at some point get turned into lactose, which is one of the main sugars that we see in milk. So then, when we're talking about our fermentable fibers or carbohydrates, those again are digested and end up getting turned into they end up getting turned into milk fat. Sorry. And then to the right is just pretty much the same thing that this graph over to the left is showing us. It paints a little bit of a prettier picture. So the rumen fermentation and digestion process, it creates energy, which then goes towards the whole process of milk synthesis. So it goes to the udders. And then, the intestines and things, as digestion is occurring in different parts of the body, it's producing or supplying the fatty acids, amino acids, vitamins, minerals, fatty acids, again, different kinds of fatty acids, to the udders, as well as protein, to the udders to create and make those milk components that then go into the secreting cells that get stored into the lumen. And when oxytocin is released, those alveoli contract and then release that milk. And so this, again, these two graphs are just basically an indication and kind of show how the feed components get turned into milk components. Okay, so when we talk about milk, we all know, I think, especially at a graduate level, that there's several different components that make up milk, especially lactose, but there's different proteins, different lipids, as well as lactose, and obviously some vitamins and minerals. I'm going to kind of go into depth about the first three, the proteins, lipids, and lactose. I'll just go ahead and make the comment now that the vitamins and minerals that we find in milk are just supplied by, the body's vitamins and minerals that the mama cow already has. And so those end up just, some of those get transferred into the milk. And so that's how that happens. But I will go into depth a little bit more about the process of proteins, lipids, and lactose being synthesized. But before I do that, I want to touch on colostrum. So colostrum, as we all know, is very important within the first 24 hours of a calf being born. And so colostrum, it accumulates in the epithelial cells before parturition happens. And so that colostrum, as we all know, is very heavily concentrated with antibodies that will eventually help immunize that calf when it's born. So that, along with building, building the immune system and getting the nutrients to that calf when it's first born or within the first 24 hours is obviously critical to its development further. So, as well as the antibodies, colostrum is also very rich in protein and has large quantities of the immunoglobulins and twice the level of the casein, beta-lactoglobulin, and alpha-lactalbumin, which we will see are some proteins as well as some fats that we're going to talk about. So, colostrum, it has, how do I say this? So, colostrum carries an increased ratio of RNA to DNA, as well as ribosomes, the endoplasmic reticulum, and the number of the mitochondria that we see per cell. And so again, this is likely due to the fact that we're just trying to jumpstart growth, continued growth in the immune system for these animals within their first 24 hours. Okay, so when we talk about proteins, I'm obviously talking about the proteins that we find in milk. So, and how those different milk components are made into the proteins that we find in milk. So the milk proteins are synthesized within the mammary glands. And however, some of the immunoglobulins and albumins can be transferred from the blood. And so as I mentioned before, these milk proteins are synthesized from amino acids, both essential and non-essential. And the uptake of free amino acids from the blood by the mammary gland can occur, as we all know, through several metabolic pathways and transport systems. So when we're thinking about, or when we're looking at the major proteins that we find in milk, there's two of them, and that's casein, which makes up about 80% of the true protein that we find in milk, and then we have a whey protein, what they refer to as a smaller extent whey protein, and that is about 20% of the true protein that we find in milk. And about 50% of the total non-protein nitrogen that we find in those proteins come from urea. Urea is one of the single largest non-protein nitrogen components, and again, it accounts for about 50% of the total non-protein nitrogen. And interestingly enough, on a side note, urea is actually one of the predominant sources of nitrogen that we use in our row crops. Not just here in South Georgia, but it is a very large component that is used in row crops across the country, and provides nitrogen to a lot of those crops. So when we think about the things that control protein synthesis, DNA is one of those things. So DNA controls protein synthesis by the fact that, or not by the fact, but so here are some of the steps that shows how DNA controls and how DNA plays a part in protein synthesis. So the messenger RNA carries encoded DNA from the nucleus to the ribosomes. Those ribosomes are in the rough endoplasmic reticulum and the cytoplasms. The ribosomes, in conjunction with the transport RNA, combines with those amino acids into peptide chains. So then the polypeptide chains are then elongated to form proteins, which then will pass and leave out of the rough endoplasmic reticulum and through the lumen, into the region of the Golgi apparatus, where they accumulate, polymerize into different milk protein molecules. So that's a little bit of how DNA. So you have DNA, or the RNA, that carries encoded DNA to the ribosomes. The ribosomes again bind with those amino acids, form peptide chains. Those peptide chains then become longer and elongate, and will then move out of the rough endoplasmic reticulum through the lumen into the Golgi apparatus. And so I think it kind of makes like this, whole process. And from there, those polypeptide chains will eventually accumulate, polymerize, and make those different protein molecules. Okay, so I really hope I'm making sense. Sorry, I have some trouble with my words. It's been a really long week, and I'm just happy to be getting through this. So, as I had mentioned previously, there's two really big major proteins that we find in milk, and that first one that accounts for about 80% of the true protein that we find in milk is that casein. So, casein proteins are characterized by an ester-bound phosphate, high proline contents, and few to no cysteine residuals. So for those of you who are really super great and absolutely love chemistry, you may know exactly what that looks like. That is not me. So some of the main types of that casein protein that we find in milk are, they're going to probably start with alpha, beta, gamma, or the kappa caseins. And so the caseins have to be phosphorylated, so bound with calcium, and stabilized by the calcium phosphate linkages and other ionic bonds before being released from the vesicles. So caseins are found in association colloids called casein micelles. So we'll kind of see where those are located within the mammary gland, I think in a few different slides. So the casein micelles include the micellar calcium phosphate that then helps with the integrity and stability of those micelles. So, which probably explains why they need to have that calcium phosphate linkage, because that just helps with the stability before it can be released from the vesicle. So casein micelles begin the aggregation process in the Golgi vesicles, or the Golgi, I think it was the Golgi apparatus within the secretory cells. So sorry, that is a typo. So within the Golgi apparatus that are within each of the secretory cells. So if there is a destabilization of these casein micelles, those can lead to a coagulation or gelation, which is how you end up with cheese or yogurt. So next, I'm going to briefly go through whey protein. So some of the major whey proteins that are found in milk are the beta-lactoglobulin and the alpha-lactobumin, which are two that I had mentioned before. And so when those are present in the Golgi apparatus, this combination, or when the alpha-lactobumin is present in the Golgi apparatus, this promotes the synthesis of lactose, which I will discuss in a few moments. So you also may see some different types of whey protein in smaller portions, and these are the serum albumin, immunoglobulins, proteose, lactoferrin, and transferrin. Okay, almost done, I promise. Not really. So then we move on to the lipids, or the fats that we know of, and which makes, everybody, a lot of people either really like or hate milk. And so what makes up milk fat is predominantly triacycloglycerols, and those are synthesized within the endoplasmic reticulum and typically are synthesized from glycerol and fatty acids. So those tricycloglycerols are formed from either, there can be short chain fatty acids that are synthesized in the mammary gland from acetate and beta hydroxybutyrate. These two, the acetate and beta hydroxybutyrate, they have their own origins from within the bloodstream. And when I say short chains, I mean the C4s through the C10s. So 4 carbons to 10 carbons, those are technically considered short chain fatty acids. So again, synthesized in the mammary gland from acetate and beta hydroxybutyrate, which come from the bloodstream. 50% of fatty acids in milk are synthesized in the mammary gland. Now, the long-chain fatty acids that are synthesized, those typically have 18 or more carbons, and they're absorbed from the blood plasma fatty acids that come from the dietary part of the cow. So based on, the nutrition that the cow has, is where those fatty acids are going to, go into the bloodstream and then be, they will be absorbed from there. And so that makes up the other 50% of the fatty acids in milk, and they are synthesized from blood plasma. So fatty acids in the mammary gland are synthesized via the malonyl coenzyme A pathway. Again, don't know if I said that pathway correctly. This is not necessarily a pathway that we have probably seen, but in a few minutes it will probably look pretty familiar to you guys. So that pathway is a metabolic pathway that converts the acetyl-CoA to the malonyl-CoA via the acetyl-CoA carboxylase enzyme. So again, we had mentioned that acetate and the beta-hydroxybutyrate is needed in order to that those two are needed to synthesize those the short chain fatty acids. And so when we think about acetate, it's one of the major carbon donors, but the beta-hydroxybutyrate, it uses the first 4 carbons of the fatty acid synthesis, but then will only continue to break off to 2 carbons as it breaks down to synthesize those short chain fatty acids. So once the fatty acetyl-CoA is formed, it will bind with glycerol or dicycloglycerol or the monoacrylglycerol to form the, I believe that's supposed to say the triacylglycerides. And so then the placement of the fatty acids on the glycerol is not random. So short chain fatty acids are essential to have a carbon in the 3 position, while the long chain fatty acids tend to either be in a 1 or 3 position. And so this is what I meant by this will probably look familiar to a lot of us. I do not believe that we have gotten too far into fatty acid synthesis, so part of this might only look familiar, but will soon look familiar. But the left part of this is what we all know and love as glycolysis through the Krebs cycle. So as we know, we go through glycolysis and make the pyruvate, which we can then make, is essential in making the acetyl-CoA. And then when I talked about that malonyl-CoA enzyme pathway, that comes from the acetyl-CoA. So that acetyl-CoA carboxylase enzyme is what helps make that malonyl-CoA enzyme, which then goes into that pathway. So I've used this picture earlier in the presentation, but it was really great to kind of discuss several different things, but this is a much better view and it's not blocked by the little recording down there on the right hand side. So again, as you can see, we've talked about the fats or the lipids and the proteins and how those go through the secretory cells, or how the milk components go through the secretory cells and end up being released into the lumen as milk components. So this is really just a better picture. So as a triacylglycerol is formed, they are rapidly incorporated into lipid-covered droplets, which is what these little blue droplets are. The droplets then migrate towards the membrane of the secretory cells, where they'll be encapsulated, pinched off, and released into the lumen. So they kind of form, they go through this process where the milk components and the fats turn them into those little lipid blobs, and then they make their way towards the membrane, the apical cell membrane, get pinched off, and then released into the lumen here as milk fat. So, lactose. One thing we all know and love in milk. So lactose, its principal function is for the regulation of water content, which helps with the osmotic content in milk. And so it is the predominant carbohydrate in milk. Some other carbohydrates that we tend to see in milk is monosaccharides, sugar, phosphates, nucleotide sugars, free, neutral, and acid oligosaccharides, some peptides and proteins, and other things like that. So lactose is the most consistent milk component, where it averages about 4.6%. There's about 4.6% of lactose within milk. And lactose is synthesized from glucose and galactose within the secreting cells. And that glucose and galactose is joined by a 1,4 carbon linkage as the beta-galactoside in order to form lactose. And then this is just a very cryptic yet funny meme that I had and found. So I do want to talk a little bit about the biosynthesis of lactose. So glucose is the primary substrate, as we all know, for lactose synthesis. And so again, as I had mentioned before, you need the glucose and galactose in order to form lactose. And so 85% of the carbon secreted in lactose is derived from blood glucose. And so the biosynthesis of lactose is initiated by the Golgi apparatus and continues in the vesicles with an influx of water and ionic constituents that cause the vesicles to swell as they pass towards the cell surface. And so if we think about the overall process and the overall equation of it, we have glucose plus this is a molecule that we all have seen, the uridine diphosphate, UDP. So it's the UDP galactose. So when that reactant is combined with glucose, it will make lactose, and the enzyme responsible for that is the lactose synthase. And so the beta-lactalbumin, which is one of the whey proteins that we talked about, has to be present in order for glucose and that UDP galactose to combine. So you need the whey protein. So we have the glucose, but you need that beta-lactalbumin, that whey protein, in order for the glucose and the UDP galactose to even combine to make that reaction possible, which forms it into lactose. And so obviously the beta-lactalbumin, which is again the whey protein, is one of the prime regulators for the lactase synthase. And so when we think about what happens in the ruminant's body, so when plasma glucose levels are low, the mammary gland blood flow becomes the primary driver for that lactose production. And again, relating it back to something that we have learned about this semester, I believe Hunter gave the presentation, but the Krebs cycle is one of the predominant metabolic pathways that generates a majority of the ATP that we use in order to synthesize lactose that we end up seeing in milk. Okay, so we're nearing an hour, and I'm going to try and wrap it up. So I guess the the big question is, why do we care? Why do we care about all this information other than passing the class? And so when we think about the dairy industry and the information that I just covered, a lot of the milk fat, milk protein, and things like that, some of those can be altered. We can change some of that through nutrition, which I'll kind of go over briefly. But these two graphs here kind of show us how we can maybe get the best out of both worlds. And so the first graph to the left shows us when protein, fat, and lactose are at their peak through the entire lactation process. So obviously, based on this chart over to the left, this line graph, we can see that fat and protein are the highest when we're looking, or when we're at the beginning of lactation as well as at the end. So it tends to, just after the beginning, kind of drop off, and then they'll kind of start to build that up. And again, that does make sense just because of the requirements that their body's needing. And when that calf is born and they begin to lactate and they're feeding that animal, a lot of their nutrition's probably going to go down for a little bit before it can start to build itself back up. And so over to the right, we will see that there's only 3 different breeds here, but we'll see how breeds can, how specific breeds have different concentrations of these milk components that we talked about. And so Holsteins tend to not have, they actually have one of the least amounts of milk fat. However, they're the ones that are most widely used predominantly because of the amount of milk that they make. However, when we're thinking about the pricing of milk can be, or is, based off of fat concentration, and there's a lot of information out there right now, and there's a lot of desire to have increased buttermilk, or butter fat content, which again you can get paid extra for. And so this kind of shows us how the time period that we're looking at through lactation can change some of those milk components, as well as how genetics can make a difference in that as well. So as I mentioned before, there are several influencing factors that can influence the components that we see in milk, whether it's the protein, carbohydrates, or the fats. So those can be environmental factors, genetics, again, as I mentioned on the previous slide, and that's evident, genetics can determine how much milk fat is present. And then obviously the health. If we have an unhealthy cow, she's going to be using a lot of her, whatever resources she has in her body, whatever nutrients she has in her body to be keeping her healthy, and probably not be producing milk. So her health is very important in her capabilities and her ability to produce good milk consistently. Nutrition, as we've seen, and kind of the basis of this whole presentation, can obviously influence the milk contents and the amount of milk contents that we see in milk. And then obviously, as we discussed, hormones is another thing, another influencing factor that goes into milk components. So nutrition is one of our biggest concerns, and obviously the easiest place for us to manipulate. So when we think about that and we think about the diet that we're feeding, dietary crude protein severely affects milk yield. And so diets tend to be higher in energy. I think it was energy digestible fermented carbohydrates. And so as opposed to undigestible fermentable carbohydrates. And so that can lead to a couple different things, one being low fat syndrome, as well as mastitis, which I will briefly talk about. So this graph here, I'll be really quick, is just, again, a graphic view of how, when we are thinking about the food that the animal is intaking and the process that it goes through in order to get to lactose and the milk proteins and the milk fats. And so this really is just a picture that kind of shows you where those processes go and how things get turned into lactose and milk protein versus the starches and things that get released as waste. So last but not least, another reason why we care about some of this stuff is mastitis and the low-fat milk syndrome that I talked about. So I'm going to briefly kind of discuss these two. So occurrences of mastitis can increase the percent of sodium as well as chloride in the milk and decrease the amount of potassium. And so I'll kind of go in a second and explain that. But basically, mastitis, I'm sure as we all know, is a bacterial infection that can result in damage to the ductal and the secretory epithelium and increased permeability of the blood capillaries. So what can happen is if some bacteria can get up through the teat or enter the bloodstream or anything like that, what can end up happening is it could end up damaging those alveoli and possibly killing half of a lobe or an entire lobe, and therefore it'll kill all of those epithelium that are on the outside of that alveoli, and then those capillaries on the outside will be able to penetrate, which is why we have a tendency to see blood in the milk when there is a case of mastitis. But going back to the sodium and the chloride and how those percentages increase, sodium and chloride are in obviously higher concentrations within the blood, which then pour into the lumen of the alveolus, and so then in order to help maintain the osmolarity, potassium starts to decrease. So again, that sodium and chloride that's in much higher concentrations within the bloodstream then are able to enter the lumen in cases of mastitis, because those epithelial are being damaged, and then osmolarity is off, so potassium goes down, and trying to help maintain that osmolarity. Calcium and phosphorus levels in milk also decline with mastitis, and this will likely reflect lower casein levels, which again will drastically alter the protein composition of milk. So again, when milk proteins decrease, blood serum proteins increase, which is again why we tend to see so much blood in the milk if there is a case of mastitis. So lastly, low-fat milk syndrome it's just a reduction in milk fat percentage, shocker, and then changes with that milk fat composition. So there tends to be an increase of carbon-18 polyunsaturated and monounsaturated acids, and decreases in some of those very long 160 carbon, 180 carbon fatty acids. And so again, that's how that milk fat composition changes. And so this tends to happen when there are changes in rumen fermentation and the availability of the endogenous fatty acid. So when we make changes to the diet and there becomes a lot more indigestible parts of the diet, then we start to affect the pH of the rumen, which obviously in turn affects the rumen fermentation. So also the low-fat milk syndrome will increase propionic acid and decrease acetic and butyric acid as a result of feeding readily fermentable carbohydrates, which depresses fiber digestion and pH within the rumen. Again, kind of what I mentioned up here. That just sounds a lot better, and so this will then obviously lead to an increased buildup of lactic acid, and then that stimulates insulin, and then will affect or affect in a negative way by reducing free fatty acid release. And so that in all again, understanding how milk is synthesized and what mastitis can do to those milk components, and how it can prevent milk synthesis is very important. So I will finish up with the citations page. I genuinely hope that you guys enjoyed my presentation. I'm very jealous that I did not end up having a pathway, because that was probably a little bit easier, but I appreciate you guys bearing with me, and I look forward to learning from the rest of you guys, and have really enjoyed the class this semester, and I really hope that you guys were able to understand some of my presentation and make sense of it all. Thank you so much.